Good afternoon. I'm Jean-Philippe David from CNRS France, and today I will talk about metabolic resistance in arbovirus vectors. And this was kind of a hard task that was given to me because it's quite a difficult subject to treat in only 15 minutes. So I will try to be rather simple, but also to give some details about the markers that you can use in the field to detect metabolic resistance. But I will only focus when data are present on the two main arbovirus vectors, Aedes aegypti and Aedes albopictus. So first, you know now from the nice previous talk that target site mutations are important in conferring resistance to pyrethroids and DDT in arbovirus vectors. But these mutations, such as KDR mutation, do not account for the entire resistance phenotype. And metabolic resistance is also it's based on the biodegradation of insecticide by detoxification enzyme. It's also important to understand that this detoxification enzyme may also sequester insecticide, avoiding them to reach the target without metabolizing them. So few facts on metabolic resistance. First, it's important to understand that metabolic resistance can affect all chemical insecticides. I mean all insecticides targeting all different physiological targets. These include insecticides that are currently used for vector controls, but other chemical insecticides also. Metabolic resistance is usually based on various detoxification enzyme families, including cytochrome P450s, glutathione S transferases, called GSTs, esterases, but also other detoxification enzyme families can be involved in metabolic resistance. Of course, metabolic resistance has a broad distribution. It has been identified in most species worldwide, including Culex, Anopheles, Aedes, and others. And it's also often combined with target site mutations in resistant populations. But studying metabolic resistance is a bit challenging because of the complexity of detoxification pathways in mosquitoes. Indeed, there is a very high diversity of detoxification enzymes in insects and specifically in mosquitoes, with sometimes more than 300 genes encoding detoxification enzymes. And also, because of the complexity of detoxification pathways, indeed, one enzyme can be involved in the detoxification of multiple different insecticides, and different detoxification enzyme could be involved in the same pathway for detoxifying a single insecticide. So it's quite a complex story. Also, the genetic mechanism underlying metabolic resistance are complex. This could be an overproduction of detoxification of one or multiple detoxification enzyme conferring insecticide resistance. But also, it's possible that the selection of particular variants of these enzymes with a higher affinity or higher ability to degrade a given insecticide can confer resistance. So, and finally, metabolic resistance is hard to track in the field, especially if we would like to go down to gene involved, because synergists and biochemists are not very specific, and we have only very few molecular markers to track metabolic resistance in the field, and most of these markers are RNA markers, which are not best suited for field studies. And finally, it's important to understand that the environment, as said by Milene previously, could have an impact on the selection and the expression of the detoxification system in mosquitoes. So this could lead to the selection of different genes in different environments, also to false positive due to presence of pollutants or the chemicals in the mosquito breeding sites. So in this talk, I will first try to give you an overview of metabolic resistance mechanisms according to the different insecticide families, and then I will try to identify key challenges for improving the monitoring and the management of metabolic resistance in arbovirus vectors. So let's start with resistance to DDT. So it has been well studied in Anopheles gambier, and it has been shown since 2003 that one particular GST, which is called GST-E2, is able to confer resistance to DDT in Anopheles gambiae and to metabolize DDT. And later on, it was shown that the same gene in Aedes aegypti was also able to dechlorinate DDT. And then, more recently, it was shown that point mutation affecting this epsilon GST can also enhance the resistance to DDT through an increased metabolism. But important to note that another expression of other detoxification enzymes, such as P450s, can also confer DDT resistance in Drosophila and Anopheles gambiae. But we're not sure yet if about the potential role of P450 in DDT resistance in Aedes aegypti and Aedes albopictus. Let's go to organophosphate. So as Dr. Veil said previously, important to know that ACE1 mutations are very unlikely in Aedes aegypti and Aedes albopictus because multiple mutation events are required. So there is probably a key role of metabolic resistance in organophosphate resistance in these two vectors. So in 2014, a specific, the overexpression of an esterase, which is called CCE-EE3E, through gene amplification was detected in a Temephos-resistant population from Thailand. And later on, the amplification of a cluster of multiple esterase genes was confirmed by next-generation sequencing. And more recently, it has been found that the same gene was also found amplified in a Greek Albopictus population showing resistance to Temephos. And functional studies done very recently confirm that this particular esterase is able to sequester and metabolize oxidized Temephos in both Aedes aegypti and Aedes albopictus. If we're now looking at what's happening with carbamates, again, ACE1 mutation very unlikely, so probably a key role of metabolic resistance to this kind of insecticide. And biochemicals usually suggest a key role of P450s, but genes involved have not yet been identified in Aedes mosquitoes. So if we are looking what's happening in Anopheles, the overexpression of P450 was associated with resistance to bendocarb. And functional studies revealed that two of them, which are called CYP6P3 and CYP6M2, are able to metabolize and sequester bendocarb in addition to other insecticides such as pyrethroids and DDT. So it's kind of concerned in terms of cross-resistance to different insecticide families. If we go to pyrethroids, of course you know that pyrethroid resistance is widespread in Aedes aegypti, with the role of P450 supported by the frequent overexpression of P450s, especially the CYP6 and CYP9 families in resistant populations. And functional studies in 2012 confirmed the capacity of multiple CYP9J to metabolize pyrethroids. And later on, other P450s, such as CYP9M6 and CYP6BB2, were also confirmed to metabolize permethrin in Aedes aegypti. Just to point out also that it has been shown that other P450s from the CYP6Z family have an indirect role in pyrethroid metabolism because they can metabolize metabolites produced by esterases. So if we look at what's happening in Albopictus, as you've seen before, pyrethroid resistance is rising quite rapidly in this species, and it's also associated with another expression of P450. Especially, it has been detected in a KDF3 resistant colony from Malaysia, and more functional studies showed that one P450, which is called CYP6P12, is able to confer resistance to deltametrin in Albopictus. So if we conclude on pyrethroids, it's clear that P450s from CYP6 and CYP9 families play a key role in metabolic resistance to pyrethroids. And there is kind of degree of overlap between Aedes aegypti and Aedes albopictus. But it's important to keep in mind that other enzymes are also involved in pyretroid degradation pathways, such as esterases, GSTs, and also probably UGTs. So now the question could be which marker to monitor, where and how. So if we look at the different markers, resistant genes that have been detected worldwide, it appears that some P450s, such as CYP6PP2 and a few CYP9Js, represent very good markers of pyretroid resistance, because they have been detected worldwide. And also GST-E2 is a very good marker of DDT resistance. And finally, the esterase gene CCE3 appears to be a very good marker of Temephos resistance, again, because it has been detected in different populations worldwide. But also keep in mind that other detoxification enzymes may also confer resistance in other populations and other environments. And finally, overexpression of these detoxification enzymes has been frequently associated with an increase in the gene copy number. This means that detection is, of course, possible from RNA, gene expression study, but may also be possible by looking at the number of copies of these genes through DNA markers, which is far more simple from field-collected mosquitoes. Just to finish, so what gaps needs to be filled in before we can improve the management of metabolic resistance in the field? So first, I believe that it's important to enlarge the panel of validated markers of metabolic resistance in main arbovirus vectors, and also to try to focus the identification of markers on DNA markers, because these kind of markers are more suitable for resistance monitoring in the field. It's also very important to quantify the relative importance of metabolic resistance alleles alone or in combination in the overall resistance phenotype, because it's clear that some resistance genes have been shown to metabolize insecticide in some degrees, but then the question is for how much it does account in the resistance phenotype. It's also very important to identify cross-resistance patterns and fitness costs associated with metabolic resistance, as this has a very important impact on the implementation of resistance management strategies. And finally, it's important to develop novel diagnostic tools for monitoring the spatio-temporal dynamics of metabolic resistance alleles in the field. And I'm sure John Vontas in his next talk will talk a bit more about that. So a lot of work to do, and I believe that for doing all this, we need to gather, expertises and skills through collaborative projects. And this is why I think international networks such as the WIN network can have a very good value for making, good outputs in this research field. Thank you very much.